Hi, thank you all for attending the end-to-end analysis of imaging mass cytometry data to study heterogeneity in the tumor microenvironment workshop. My name is Mark Sadie, and I am really excited to have you all here as attendees for this workshop. Ultimately, I'd like to present essentially a framework for analyzing multiplex data. Now, of course, I'll be using imaging mass cytometry as an example, as evident by the title, but by all means, you could potentially even apply this to Codex, which is now rebranded as PhenoCycler, or simply, 4 or 5 channel immunofluorescence data. So on that note, let's get started. So just a quick overview of the workshop. Foremost, I'd like to, present the biological question leading to the development of this analysis pipeline. No method is really developed out of the blue without any kind of context or why it was made to begin with. And so I'll be giving a little bit of background for this project and how it kind of ties into the research foci of the lab that I'm a part of. Next, I'll focus on what the extent of image analysis is within QPath. So that'll include things like cell segmentation, cell classification, regional segmentation, and then finally data visualization in Python. So you can essentially evaluate things such as marker intensity distributions, marker-marker colocalizations, percent positive scoring, and even dimensionality reduction to identify these sorts of phenotypic similarities between various cases in your data set. The second part of the workshop, essentially at the end of this presentation, will be a practical demonstration. As the title suggests, this is meant to be an end-to-end workflow. So I'm going to start off with the typical MCD or text files that are available from the Hyperion system, and ending with statistics and figures that one could potentially use in the publication. Now, there will be a wide variety of imaging mass cytometry projects, or let alone multiplexed imaging projects out there, and some methods of visualization might not be applicable, others might need a little bit more elaboration. So what I really want you to take away from this is the framework and the sort of modular aspect of which you can slice and dice according to your own needs. So just to give a little bit of background on imaging mass cytometry, it's a technology where tissue sections are stained with heavy metal conjugated antibodies. These are then ablated and imaged using a Cytometry mass cytometer, which effectively allows for highly multiplexed imaging without limitations conferred by spectral overlap. One of the reasons why we can't have 30 fluorescent markers on a single tissue section imaged at the exact same time. By overcoming the limitations associated with spectral overlap, this essentially allows for high-plex or multiplexed imaging modalities to exist, such as, but not limited to, imaging mass cytometry, Codex, MIPI, and the list goes on, digital spatial profiling. So my kind of research focus essentially revolves around tumor oxygenation, or rather the lack of tumor oxygenation. Tumor hypoxia essentially refers to the low level of oxygen present in some regions of tumors. And oxygen levels and radiation sensitivity decrease distal or further away to perfused vasculature. So one of the main reasons of a biological importance to oxygen is the fact that it's needed for effective radiation therapy. If you have hypoxia, well, you have cells that are now resistant to radiotherapy, as well as several chemotherapeutics that target aspects associated with hypoxia, such as cell proliferation, which is negatively correlated. So in a nutshell, hypoxia confers biological changes to the tumor, such as increased aggressiveness, therapeutic resistance, and increased metastasis. So as part of this project, we essentially wanted to identify potential biomarkers of hypoxia. We have several patient-derived cases which were given a hypoxia probe called thiaminidazole, which effectively is a relatively non-toxic drug that binds irreversibly to regions of low oxygen. When this drug binds irreversibly to regions of low oxygen, it can be detected using traditional immunohistochemistry approaches, such as HDAB staining or immunofluorescence staining, but in this case, imaging mass cytometry. So it can effectively identify regions of relative hypoxia and relative normoxia based on the spatial and intensity distribution of this marker. Our goal here is essentially to perform a sort of differential expression analysis, comparing the relative expression levels of all of our markers, all 28 or so markers in our IMC panel, relative to hypoxia, as ultimately this can tell us whether or not certain markers are correlated with hypoxia and would potentially serve as a reasonable biomarker of hypoxia. So I might throw around a lot of hypoxia terminology, but by all means, it's probably not the most important thing relative to this presentation. What I really want to essentially outline is aspects of this pipeline that would be applicable to your project. And so one aspect that I would say is applicable to a lot of projects involves cell segmentation and classification. So those are what I will be performing, the bulk of my time during this workshop, essentially discussing and elaborating on. So as a first step in this workflow, we wanted to identify regions of hypoxia, but these could be regions of other structures in your tissue, such as tumor versus stroma, or anything you could essentially elucidate in imaging mass cytometry datasets that you might have. So we first essentially applied a pixel classifier consisting simply of a slight Gaussian blur and a binary threshold for classifying regions of hypoxia shown in green and regions of relative normoxia shown in red. I'll be abbreviating these as PIMO positive for the hypoxic area and PIMO negative as the relatively normoxic area. Next up, we perform cell segmentation. Now, cell segmentation, much like the pixel classifier, is another tool available in the vanilla or unmodified version of Keypad. However, imaging mass cytometry has a relatively poor spatial resolution of about 1 micrometer per pixel, which is roughly 4 to 5 times lower than that of a 20x fluorescent image. So I'll be using something I partially developed called Universal Stardust for QPath, which essentially allows us to apply a pre-trained machine learning model to segment this image. Next up, we want to, ultimately classify our cells. So one key difference about this pipeline compared to existing pipelines, such as the one developed by the Bodenmiller lab or what Codex might develop as part of their Codex Map platform, is that I actually don't use dimensionality reduction followed by clustering to identify cell populations. Rather, I use a pathologist. I have a meeting with a pathologist and we sit down for each and every one of these markers to identify some sort of binary threshold for classifying a cell as either positive or negative for this marker. We then repeat this for each of the markers included in the panel, and I'll essentially elaborate more on this during the actual practical part of this workshop. So now that we've essentially created our sequential classifiers for each of the 28 different protein-based histological markers, we can then construct a composite classifier, which assigns one or more class to every single cell in the image corresponding to a list of markers that this cell is positive for. So as you can see by the yellow cell that I've highlighted a bit in the center, you can see that it's positive for markers such as the trimethylhistone, the DNA intercalating agent, lactate dehydrogenase, and the list goes on. Next up, well, we want to essentially determine whether or not these markers were being enriched in a hypoxic PIMO positive or normoxic PIMO negative area. We essentially compute the fraction of cells positive in each of those areas for each single kind of marker classification. And this ultimately tells us, canonical HIF targets or known hypoxia regulated genes such as CA9, LDHA, and HK2 are all, overexpressed in the hypoxic areas. Now, this is very unlikely to be a directly, a crucial part of your own project. And what I really want to outline is we can identify regions within an image and compare scores of various marker intensities across all of these different marker categories across both regions of an image. So ultimately, it depends on how you want to segment regions in your image, if any. You might not have any regions. You might simply want to quantify all cells within all ROIs from a certain grouping. And again, I'll show that a little bit downstream in the analysis. So by all means, no need to get held up on this image pixel classifier segmentation if you ultimately have no need for it. Next up, we can essentially compare marker intensity distributions. So violin plots are nothing more than a sideways histogram with a box plot overlaid onto them. But what this can tell us is whether or not there is a statistically significant difference of cells' markers' intensity across two or more different groupings. Now, in this case, we pooled all of the PIMO-negative areas and all of the PIMO-positive areas from all images in our batch, and we essentially wanted to determine whether or not the distribution in intensities tended to differ. However, the grouping again can be done on more of a, I guess you could say, macroscopic scale, in which we can group by certain patient metadata, such as that relating to a control group or experimental group cohort, or even something such as mutation status of a given sample. Next up, we can also determine marker-to-marker correlations. So we can compute the Spearman's correlation coefficient to determine whether or not two markers are correlated with one another. As expected, a lot of the known hypoxia targets, such as LDHA, CA9, and GLUT1, have a strong correlation with what is effectively a ground truth of hypoxia, the PIMO stain. And interestingly enough, markers such as vasculature, CD31, or markers associated with vasculature, such as alpha SMA, typically have a negative correlation with hypoxia. But again, this is a great way of simply determining whether or not any two markers in your panel of antibodies are correlated with one another. And finally, we can perform a little bit of dimensionality reduction. So let's say we have 28 different markers in this panel. Well, we're going to have about 4 different measurements corresponding to the distinct cellular subcompartments, multiplied by 5 different descriptive statistics relating to those measurements. And you might end up with a per cell statistic sheet consisting of thousands of different features. Dimensionality reduction techniques such as UMAP, t-SNE, or even PCA are a great way of viewing high dimensionality data sets to essentially identify points in your data sets that share phenotypic similarities. In this example, I've colored the data points based on individual samples or patients that they came from. And we can essentially use this to identify whether or not two patients share some kind of phenotypic similarity or dissimilarities to one another. Of course, we can also color them based on the marker intensity present. So, if we do end up seeing differences, well, we can figure out what markers visually are either overexpressed or underexpressed according to these different groupings. So, in a nutshell, we essentially provide an end-to-end workflow for analyzing hypoxia-dependent expression in imaging mass cytometry images. But this is generally robust enough that it doesn't have to do anything. It doesn't need anything to do with hypoxia or even imaging mass cytometry data. The framework here is simply cell segmentation, cell classification, regional segmentation if needed, and grouping of the data points based on some kind of categorical variable. So now I'll quickly turn over to the practical session where I will essentially demonstrate how I would go about analyzing this data. So at the time of the conference, I'll be making publicly available a subset of our IMC data consisting of anonymized samples from 2 cases, 3 regions of interest from each of them. I'll also be showing, well, as the name suggests, an end-to-end workflow. So we'll start off with a fresh QPath project. I'll be using QPath 0.4.0 or a snapshot of that, but it is more similar to the official release of 0.3.2. I'll also be using some classifiers that I've made ahead of time simply to save space. I mean, once I've developed or showcased how to create 1 or 2 classifiers to classify individual cells, there isn't really any need to, go through all 28 of the different markers simultaneously. And as I mentioned, the recording, code, and sample images will all be available. So by all means, follow along at your own pace. You can watch this recording at your own time, so you can pause and play it whenever you need. So there's no rush to keep up. If you're watching this presentation live, what I really want you guys to take away from is simply just the framework, not necessarily following along step by step. It's also worth noting that the machine I'm running at the moment has 120 gigabytes of RAM with 32 threads of a processor. So things might go a lot faster than they would on, say, a laptop. So yeah, but coincidentally, it actually does work on a laptop. I've been able to replicate this pipeline on my old 5-year-old Alienware laptop. So there isn't any kind of computational necessity requiring execution of this pipeline or anything demonstrated here. Yeah, so on that note, let's begin the practical session. All right, so welcome to the practical session, or rather practical portion of this workshop session. As I mentioned earlier, there's no need to, keep up in real time with this presentation. I'll make all of these recordings publicly available. Images will be available, source code will be available in both the Groovy scripts and Python scripts. So, feel free to follow along at your own time. So let's talk about prerequisites. What do you need prior to actually starting this pipeline? As seen on screen, you need QPath. What version of QPath, you might ask? I'm guessing you asked. 0.2.3 or later. Realistically, there isn't much of a difference between any of the newer versions of QPath, with the exception of the set of libraries that are actually being imported when I'm running the Universal Stardust segmentation script. But I'll get more into that once we actually reach that stage. So for now, you can run this pipeline on 0.2.3, 0.3.2, or 0.4.0 and any snapshots. In this case, I'm running a particular snapshot built back in early January. Only difference is I've built it with some CUDA libraries just for the GPU accelerated cell segmentation. And yeah, so then what you'll need next is Python, or if you're a Python programmer, maybe some kind of Python interactive development environment. My choice here is Spider, but there are tons of Python environments out there for, kind of user-friendly programming. So I'll be using Spider later on for when you've actually exported the data out of QPath, but you can use any kind of Python development environment, or even R. There isn't any reason for why you can't simply use R or MATLAB or some other programming language for statistical analysis or data visualization. The scripts I'll provide are just a demonstration of the different ways you can kind of slice and dice your data, but are by all means not an exhaustive list of what can be done with a data set as complex and multidimensional as an imaging mass cytometry data set. And finally, what we'll need is a way of getting IMC data into QPath. So a while back, when I was, just browsing the forums, I noticed that there wasn't a lot of posts revolving around multiplexed image analysis in QPath. I mean, there's been some excellent extensions and plugins developed by various forum members, such as Mike Nelson's Cytomap and Back, which is a great way of essentially taking cluster-based classifications into QPath to assign sub-classifications to. But I just wasn't seeing a lot of, IMC analysis or codex analysis. And one of the reasons being is that the currently available methods for exporting images, Hyperion images, in a way that is readable by QPath as some, let's say, technical issues. So let's take a look at one of these forum posts. There's a forum post a while back where a user was trying to load in a 38-channel IMC image. Now, one of the packages that comes with the IMC workflow is the MCD viewer, and the MCD viewer will allow you to export OMI TIFFs. Of course, OMI TIFFs can be read by QPath. Now, the catch here is that OMI TIFFs generated from MCD viewer have a small bug, at least dating back to November 21st, which is when I last tried it out. It incorrectly specifies the channel metadata, and so when you try and load in an image, you'll get just a bunch of fuzzy noise. Now, STAR, which is a core facility located in the University Health Network of Canada, actually developed a IMC converter. This essentially converts the text file or MCD files generated from the Hyperion imaging system into either single channel TIFFs, or in our case, OMI TIFFs. And again, they've outlined that there are certain issues with Fluidigm's official MCD viewer, which, hey, I mean, by the time of this presentation might have been fixed. So if it works and you have no problem loading OMI TIFFs generated from MCD viewer, by all means, use them. But we encountered this issue. Star helped develop this script, and all this stuff is publicly available. This isn't like locked away behind a private repository or anything. So I'll post a link to this in the video description, or you can just search Star IMC converter or check out this forum post titled Cell Segmentation in QPath with Multiple Images. So you can either have this run as a standalone executable or through Python. Here I'll I've downloaded the standalone executable. Now, unfortunately, I don't have the original MCD or text files which were used to generate the OMI TIFFs, but all you would really do is basically pop in some kind of directory for images that you want to convert and an output directory for where you want your OMI TIFFs to be saved. You can then put a little bit of additional arguments, such as what to assign for missing data or compression. And here I believe they use some kind of LZW-based lossless compression, so it simply just cuts down on file size quite dramatically, actually. I think it reduces file size by about 10-fold compared to the original text files that have no compression. Anyways, once we've run that IMC converter, we should have a set of images. I can find this as located here. So these images are what I'll be making publicly available. And again, I've been, de-identified. There's no patient information in these images, but they are of human glioblastoma. So they'll become publicly available. Anyone can download them. They're fairly small, despite having close to 20 different channels. And yeah, let's go ahead and load it. So first thing we need to do is create a QPath project, just as we would with any sort of analysis in QPath. I've made a little empty folder already here called demo project. So let's go ahead and create a project here. I will also turn on input display. So now anytime I do a left click, a right click, up, down, right, left, it'll kind of show up here. So if I'm doing any weird shortcuts like Control S or Alt Tab, you don't have to wonder what buttons I was pressing. So yeah, let's go ahead and create ourselves a project. Paste a directory here, select the folder. Now we have an empty project. Next thing we want to do is populate the project with images. So let's find our images. There's a couple ways to go about this, but honestly, just dragging and dropping is the go-to way, at least for me. And that's set the image type to other. I don't think it makes that big of a difference for any of the downstream analysis, but hey, it's the closest I can get for IMC data. Auto-generated pyramids can be checked, but again, these images are no larger than about 1,000 by 1,000 pixels, so I wouldn't expect a lot of pyramid levels to be generated anyways. And yeah, let's go ahead and import them into Keypad. So great, we've got a project consisting of 6 images, 3 ROIs from 2 different samples. Now, of course, we can always, open an image, go through each and every one of the markers one at a time and do quality control to make sure that the staining looks right and that the image looks right. But when you're working with IMC batches, you might have hundreds, if not thousands, of IMC ROIs being acquired. And so it no longer becomes feasible to go through all 28 channels of these hundreds of ROIs one at a time. What I like to do is to stitch a few representative ROIs side by side and have them all be shown on the screen at the same time. So how can you do this? Well, thanks to the ability to create sparse images in Keypad. So sparse images basically a in-memory, I guess you could say, image consisting of several subsets of images all stitched together side by side. And one way we can do this is by creating these training regions of interest. So we can go to Analyze, maybe Objects, Annotations, Specify Annotation. So now we can create a rectangle of classification. Now, actually, let me reset the classifications because these actually carry over from previous projects. Classification of region, sure. We'll make it 1,000 by 1,000 pixels. We add an annotation, and we see it nicely overlaps around the border of the entire image. In this case, each IMC ROI is 1,000 by 1,000 pixels or 1,000 by 1,000 microns. But you might have IMC images that are larger or smaller. So as a result, you can adjust the size of these ROIs accordingly. So now let's move on to the next one. We add an ROI, move on to the next, and so on and so forth. Now, for a project with about 143 images, it's not really feasible to do it for all of them. So you might want to just do it for a small subset of images. Here, I've essentially done it for 4 out of the 6 images. And now that we've created region annotations for 4 out of the 6 images, let's stitch them side by side. So we can go ahead and go to let's see, where would this be under training images? We would create a training image. Because we have 4 images and we know that the side length of an image is 1,000 pixels, why don't we make it, 2,000 by 2,000? So preferred image width to 2,000 pixels, and we press OK. So I believe it is stitching it right now. And great. So this will essentially serve as the image in which we want to optimize our thresholds and various parameters. So what is the first thing that we'd want to do? Well, if you recall from the presentation, we wanted to develop a pixel classifier to identify regions of hypoxia. So we can, well, first turn on the channel corresponding to our marker of hypoxia. This would be pymonitis, or pymo for short. And now we can essentially create a pixel classifier. In this case, I prefer using a thresholder at relatively low resolution. We would use the pymo channel, or whatever channel it is that you'd want to use for segmenting different structures in your IMC image. We then apply Gaussian pre-filter, a little bit of smoothing, set some kind of arbitrary threshold. Again, all this would be done, with someone who has experience in these images, because some people might not know what tumor versus stroma delineations are, ideal, and you might need help with that from a pathologist or someone with some experience in that field. Now, because I've actually created this thresholder before, I'll simply load in one of the pre-existing classifiers. Let me just pull that in. And all right, just gotta figure out which one to use. So if you ever want to load in classifiers, you can simply open up the project directory, open the classifiers folder, and just copy and paste an existing classifier. In this case, I wanted to use the following classifier, which essentially tells me the binary threshold used and the extent of Gaussian blur. You can also use machine learning approaches to essentially train your own classifier, and that's perfectly acceptable. One key part of this pipeline is that it's highly modular, so you can easily substitute any part of this workflow. So on that note, let's go ahead and apply our classifier. We load a pixel classifier. I might not have import from files. There we go. So let's go ahead and create ourselves some objects. Of course, the parent objects will be full image. The object type will be annotations. You can also specify what you want your minimum object size and hole size is, but again, we're not going to worry about fine-tuning those parameters until, well, not for the sake of this demonstration. And yeah, let's go ahead and create those. So one thing to note is that if you created classifiers from scratch, you would have actually had those classifiers exist ahead of time, or classes exist ahead of time. So let's quickly just populate that from all classes. Now we have our pymo negative, pymo positive class. I'm just going to make pymo negative in red and pymo positive in green, simply because I'm a fan of this way of visualizing it. Next up, cell detection. So we're going to quickly take a small detour and switch over to our DNA counter stain. Now, for cell detection, we can either process our entire kind of training image, we can process a small subset area. For the sake of brevity, I'll just be drawing a small arbitrary rectangular area, and here's where we'll actually try out our cell segmentation. So Keypad, has its own way for cell detection. It's a traditional watershed segmentation. We'll use the DNA counter stain, which is known as DNA 193. We could also use DNA 191, two different isotopes, but from the same probe. Threshold, well, we can always hover over a typical positive cell, and we see that maybe a threshold of, say, I don't know, 50. And Yeah, let's just give it a first pass, see how well it works. So does a half decent job. I'm not super excited due to challenges of segmenting these areas. We can always bump down the threshold and repeat this. But then again, watershed segmentation, given the poor resolution of IMC and how you might have some not so good looking staining, you might want to opt for a different method of cell segmentation. One that might be a little bit more robust, one that might have been trained on a larger set of images. And this is basically a great point to kind of, I guess you could say, shoehorn in universal Stardust for QPath. So what is universal Stardust for QPath? It is essentially a pre-trained machine learning model that you can now apply to segmenting IMC data amongst a wide variety of other modalities of data. So QPath has a lot of support for using Stardust, whether it be for applying pre-trained models or even training your own models. There's a lot that QPath can do in terms of using this relatively good quality, I guess you could say, method of cell segmentation. So there is a little bit of installation required to get this all set up. I mean, it's all really outlined in, well, not only in this GitHub repository that's publicly available, but on the Stardust, on the QPath documentation. So it explains how you're able to obtain these pre-trained models, as well as, tuning your own parameters. What universal Stardust for QPath really does is it packages all of this in a nice, slightly more kind of user-friendly method, instead of you trying to figure out, which kind of pre-processing steps are needed to essentially apply a IF trained model to segment IMC data. Because if you keep in mind, the dynamic range of IMC is going to be drastically different from that of traditional fluorescence data. So on that note, let us load a Stardust segmentation algorithm. So I have a copy of Stardust already downloaded, and I will quickly load up that script. Let's see. Multimodal Stardust. Actually, I'll use the GPU accelerated version just to speed things up very slightly. So let's take a look at some of the parameters we might need to change. In your case, you have to specify where is your pre-trained model located. I like using the DSP 2018 heavy augment model, but I believe there's 2 or 3 that are publicly available that can be applied to IMC data. You also have to specify what channel to use for nucleus detection. So let's take a quick look. Our DNA intercalating agent is channel 22. So we'll set this to channel 22. You can apply a little bit of median blur. You can apply simply divide all pixel intensities, add a value to all pixel intensities. Here's the threshold for detecting. Many different parameters you can play around with. But for now, let's just go ahead and see how well it does out of the box. So we'll run it. Unfortunately, I just realized it has select all annotations being highlighted. So instead of just analyzing this small little rectangular area, it's going to attempt to perform Stardust segmentation on all cells of this image, operating at the full image resolution. So yeah, thankfully it didn't actually take as long as expected. But you can see it does, a half decent job of segmentation. Let me actually delete this object. Just delete this. Nope. And great, now we've got segmented cells. So of course, this still doesn't seem ideal. We can go back and forth trying to tune the parameters until we get something that meets our expectations. Because I've actually processed these images before, I can try and load up what our current segmentation script ended up being, although I believe it might have been an older version. Anyways, let's copy that script, paste it into our scripts folder of the project. Make a new folder called scripts. And I have a feeling this will probably give me an error because I'm using a script meant for an older version of QPath. But let's give it a shot. Yeah, hopefully it works. Okay, apparently it runs, surprisingly. Another thing to note is this little line over here clears any existing detections. So if you did end up performing any method of object detection ahead of stream, you might want to comment this out. Anyways, we will wait for this to finish running. Hopefully it doesn't crash, but I don't see why it would. And there we go. Well, that is a bizarre situation. Okay. I believe it actually processes one region of interest at a time. Another thing to keep in mind is I've actually set the cell expansion radius to be a bit larger than expected or than needed, but this is kind of what we used in this current project. So yeah, now we essentially have segmented cells, or not so much segmented cells, but rather an image containing the ideal parameters for segmented cells. So let's go ahead and save Composite Classifier v1. Well, this actually leads me on to the next step, which is creating the Composite Classifier. So let's talk about cell classification. One unique part of this pipeline is that it actually doesn't use dimensionality reduction followed by clustering as a means of identifying distinct cell populations, unlike what's conventionally done in the Bodemiller pipeline or what Akoya Biosciences does with their Codex pipeline. Instead, we sit down with a pathologist and we create an object classifier. This object classifier essentially is a single measurement classifier, where for every single detection, we will work on each of the different protein coding markers. We'll figure out where it's being localized in this case, alpha SMA cell mean. What we also need to do is populate all classes, populate from image channels. So now we have one class corresponding to each of the image channels, which we can then assign the above threshold class to now become. So now if we switch over to the alpha SMA positive channel, or sorry, alpha SMA channel, we have some kind of level of threshold being set. Now, by default, I believe QPath uses the mean plus standard deviation, but sometimes you might want to raise or lower this threshold. So it's simple enough to just drag and adjust the slider until you're correct in identifying the cells that you believe to be positive for this given marker. One thing I like to do is actually hide the negative cells, just because they're not really of interest, and we can actually hide these classes in the viewer. So now when we're going ahead and we're fine-tuning our threshold, we get a little bit of an easier way of visualizing it. So then we will save the classifier using the name that it automatically generates corresponding to the image channel name. And we click save. And then we move on to the next one and the next one and so on and so forth. Effectively, we should now have a project containing a set of cell classifiers. And what I'll actually do is I will load up our object classifiers that I previously generated. So I believe all I have to do is copy and paste them, if I'm not mistaken. Object classifier. So let's open the project-based directory. Go to classifiers. My bad. And create a new folder called object classifiers. Within this object classifiers folder, I'm just going to copy and paste the ones I've already created. But again, they were all created in the same manner as shown previously. So you can see I have one classifier for each of the different protein coding markers, including the panel. I also tossed in the DNA counter stains because why not? You know, there's no harm in having those. The one exception here is this little composite classifier, and the composite classifier is nothing more than a set of object classifiers being applied sequentially. So the way this composite classifier was created was by going to create composite classifier, selecting all of the classifiers, and we call it composite classifier v1. So at that point, we would click save and potentially even save and apply. But because we've already done that in a previous project, and because I don't want to trouble you with having to sit through me going one at a time, creating the appropriate threshold for each of the protein coding markers, we can just go ahead and load the composite classifier v1. So now we have this beautiful image that is extremely challenging to interpret, visually speaking, because now we've assigned a cell classification for every single cell in the image corresponding to a list of markers that it is positive for. So if you see this little yellow cell over here, it is positive for quite a lot of markers. The histone marker, the DNA counter stain, LDHA, CD68, GFAP. So, you know, visually, this makes a great picture. It shows the heterogeneity in this tumor microenvironment. Any cell colored the exact same color as another cell means it's positive for the same set of markers. And we can actually see, you know, a little bit of similarities between the cells that are being positive in this hypoxic area or cells adjacent to vasculature. We also see regions outside of the hypoxic areas that seem to also be positive for our hypoxia markers, which are, you know, equally as interesting. Now, on that note, we have created a pixel classifier for object segmentation. We have created a classifier for cell segmentation and for cell classification, but we haven't actually applied it to any of our other images. So, in other words, now that we've optimized our parameters and our thresholds, what do we do from here? How do we apply it to all these images? So if you go back to the workflow tab, you can actually click create script. And this will basically create a script with a list of actions that were performed. Now, not all of the actions were actually relevant. In this case, we don't need to perform these watershed cell segmentations because we opt for our own method of cell segmentation. We do have to set the image type. We might want to clear all image objects. So I believe to clear all image objects, we can go delete all objects. You know, let's go to a blank image. Delete all objects. And even though nothing seems to have happened, Let me just, there we go. Objects, delete all objects. There we go. It is now populated as clear all objects. So let's actually put this up here. Clear all objects. Always better to start fresh when you're analyzing a new image. We do want to load up our pixel classifier. No need to clear detection because we don't have any. Don't need to clear the selected objects. We do have to perform cell segmentation before we actually run the object classifier. And so all of this can be done by simply copying and pasting this. Not the ideal way of essentially injecting it into this pipeline, but if we want to have one script that serves as an end-to-end workflow, at least in QPath, presumably we'd have to copy and paste the Stardust segmentation and paste it inside of our existing kind of end-to-end workflow script, where the prerequisites are regional segmentation and the post-processing is simply a object classifier. So let's see what happens. Might work well the first time out of the box, might give us some errors. Who knows? Let's see how this runs. So you can see already in the background, it's segmented these distinct areas, perform the cell segmentation, perform the cell classification, and seems to have gone well. We can go ahead and save this and now run it for all projects. Well, except for the ones we've already run it for. And let's give it a second to run. Should take about as long as processing the 4 stitched tiles side by side. I mean, as you can see, it's already completed 1 out of 4 images. Oh, sorry, 3 out of 5. 2 more remain. And we are nearing completion. Great. I'll also make a habit of, just saving this script as maybe demo workflow within the project's script folder. So let's quickly take a look at some of the other images, just, better to make sure it actually ran. Yep, doing a great job. If we look at the cells, we'll see, I can actually make this a little wider. We now know a list of markers that the cell is positive for. We know whether or not the cell falls within a PIMO-positive, hypoxic, or PIMO-negative area. And now we have tons and tons of measurements relating to mean cell intensity or median, min, max in the cytoplasm membrane cell for each of the 28 different protein coding markers present in this panel, as well as some shape features. And again, if you're not satisfied with the shape features, you can always, calculate some more shape features. Or maybe you want to toss in a little bit of spatial analysis down the line. If you do distance to annotations, you can compute the distance of every single cell centroid to the nearest annotation border. We can also detect centroid distances to compute the distance of every single cell to another cell, essentially telling us whether or not 2 cells of a given classification are in close proximity to one another. But for the sake of this demonstration, I'm going to leave out any kind of spatial analysis because the demands, or rather interests, I assume the attendees you guys might have can be drastically different from what I've had. In my case, I was interested to see how marker intensity distributions change as a function of distance to the nearest blood vessel. If you recall, we have various markers relating to the blood vessel included in this panel, but what I haven't shown is how to segment vasculature, how to compute distance to the nearest blood vessel, and various other methods of data visualization relating to spatial proximity of cells to the nearest decimal. The reason I haven't shown that is because I want to make this pipeline, or rather this demonstration of this pipeline, relatively high level, easy to interpret, easy to implement for your own projects without having to worry about all this kind of spatial complexity. That being said, I will be releasing, or I will be co-releasing a paper with a co-author. I'm a co-first author of the analysis pipeline applied to these samples, in which we do actually use some of the spatial features for studying the tumor biology. So I hope to get this paper at least released in the coming months, and if possible, I'll provide a link to the publication in the description of this video, essentially outlining the full extent of this pipeline and how I've applied it to my own work. And again, all the code for the official release will be publicly available as part of the publication. So, feel free to use that. Anyways, now that we've applied it to our images in our data set, let's kind of begin to wrap up the QPath side of this workflow, in that we need to finally export our measurements. So there are two types of measurements we'd want exported for all images except for a little sparse image. We want annotation level measurements, preferably as a CSV file. And we can call this annotation measurements. And we will export that. And then we will do cell level measurements. And we can go ahead and export that. Cell measurements. And these will be the two main measurement files that we will use in any kind of downstream analysis. So let's go ahead and export those. Might take a bit longer for the cell detections now that we have, thousands of cell detections per image, rather than just having two regions or two objects per image in terms of annotations. But on that note, I believe this now wraps up the QPath portion of the workflow. So let's just confirm that the export was successful. Yep, seems to have worked. And cell measurements. I might have regretted opening this in Excel. Great. So on that note, I believe this concludes the QPath portion of the workshop. And now we'll be working on data visualization and subsequent statistical analysis in Python. Right, so now we are reaching the end of the workshop, in which we will be focusing on data visualization in Python, as well as some statistical analysis. In the interest of time, I've gone ahead and generated, the majority of figures, which will be outputted from the following three scripts. However, I'll still take the time and walk you through each of the code blocks and fragments of these scripts and what exactly they do, what do they generate. But ultimately, you might be using some or all, or even none of these to visualize the data. This portion of data visualization is highly dependent on what you're exactly looking for in terms of your IMC or other multiplexed data set. In the case of tumor oxygenation, our ultimate kind of goal for this project was to identify what genes are differentially expressed in response to variable oxygen concentrations. And so a lot of the considerations in terms of how the data was being partitioned, grouped, how double positive scores, single positive scores are being calculated, all of this stuff was essentially done with that in mind, that we're trying to essentially identify drivers of or rather genes that are upregulated or downregulated under hypoxia. So depending on what needs you have in your own project and how you've constructed your panel in hopes of assessing difference in microenvironmental heterogeneity or whatever aspect of that, this script might be very relevant to you. This might be not at all relevant to what you're interested in. But again, this is just to illustrate some methods of ways you can analyze and visualize data generated from QPath. So we last left off generating these exports from QPath, the annotation level export, where we have multiple measurements corresponding to every single annotated region. In other words, a pymo-positive and a pymo-negative area per image, or the cell measurements, where every single row corresponds to a single cell and every single column corresponds to a different measurement relating to that cell, including various categorical variables, such as a list of markers that the cell is positive for. For the bulk of the analysis, we will only be using the cell measurements, or rather a processed version of the cell measurements file. But equal, or rather not equal, but comparable levels of analysis can be done through annotation level measurements or any other export from QPath. So first things first, we want to append categorical information that will be used to group cells as belonging to a patient that came from an experimental control, or maybe a primary or wild type tumor, or what have you, whatever patient metadata. Now, since these samples are effectively anonymized, there isn't an actual biological grouping I can show you other than the fact that sample A is now labeled as this control group and sample B is now labeled as this experimental group. But as I mentioned over here, these are just arbitrary groups. They have no biological meaning, and they just illustrate how you can essentially append supplementary metadata to this data set from external sources. So on that note, let's load the various libraries we would use. As you can probably tell, we don't end up using most of these libraries, given that we're not computing Spearman correlation coefficients. But all we're really doing is just adding 2 extra columns, or rather 3 extra columns, a column corresponding to what group the patient came from, and 2 other columns corresponding to the IMC ROI and what patient that or what sample this ROI came from. So on that note, let's run the scripts. We've just loaded libraries. We're specifying where is the data that we want to read. So it's our cell measurements file, and what the output path is, the process cell measurements file. You can have output path be equal to CSV path, but I don't recommend it because then you risk overwriting data. And if you do a mistake, it's that has to be re-exported from QPath. So now that we've kind of declared what we need to declare, where we're going to read from and where we're going to write our file from, we can specify what kind of metadata we want to include and append new columns to the data frame. And finally, write it out to a new CSV. So if we take a look at this demo project, the moment I run this script, or rather the last line of this script, we will have a new file being generated, processed cell measurements. Same as this, just an extra few columns appended on. Now we can do the same for the annotation level measurements, even though we don't really use it. All we'd have to do is simply change it from from cell measurements to annotation measurements, and just run the entire script again. Done. So now we have processed annotation measurements and processed cell measurements, which effectively contain one added layer of information that may or may not be useful for subsequent data visualization. Now, this script is basically a smorgasbord of cobbled together figures, statistical analysis, essentially a series of different types of ways of slicing and dicing and reanalyzing the data to meet whatever it is that I was aiming to analyze at the time. So you might be using some portions of this, some code blocks, others might not, others would use all. Again, this is just to illustrate what can really be done with the exported data. So first thing we want to do is, of course, load in our data, which is effectively done during this code block. It would be nice to rename some of the columns if we want to view them in a different manner when we're essentially, say, labeling X ticks or Y ticks in certain plots. So we can go ahead and do that at the start, right after it's being loaded in. Next up, we have to set some constants. Now, I can't really go into a lot of detail about what each constant does until I actually encounter use of that constant down the line. But this is basically a way of, saying what columns we would want to pass into the subsequent functions for plotting the data. This is used to establish various p thresholds for Spearman correlation coefficients, such that maybe we'd want to add a single asterisk or 2 asterisks or 3 asterisks on certain plots to illustrate statistical significance in terms of correlation. Things like this seed would be used as a constant for UMAP plots or other dimensionality reductions to have them be a little bit more reproducible from run to run. But one of the most important variables, I would say, is this, measurements of interest. This is essentially a list containing what measurements we are using to classify cells. So if you recall earlier on, we were creating a composite classifier by essentially sandwiching multiple single measurement classifiers together. The measurement that we used for the single measurement classifier is what we would use in the measurement of interest. So for example, when we were classifying cells as being alpha estimate positive or negative, we were using the measurement alpha estimate positive cell mean. However, when we're using things like the like a protein that is exclusively expressed in the nucleus, such as K67, maybe we're using K67 nucleus mean. So as a result, we'd like to append that as part of the measurements of interest. We can also drop certain measurements of interest that we're not really interested in plotting, such as the DNA counter stains, again, because we don't really expect them to differ all that much. Yeah. And of course, if we want to filter our data set prior to any analysis, just to focus on, say, specific group, maybe only analyze control samples independently from the data set, we could do this here. Of course, currently it's commented out. This operation essentially gets unique IHC marker names. If you take a look at the marker short variable, it contains a list of 18 different markers essentially derived from their channel names, or rather their measurement names, which are derived from their channel names. So, this is probably what we would want to show on a plot when we're trying to label things LDHA or SOX2, rather than the full channel name that contains the heavy metal tag, LDHA, and a bunch of other characters that aren't super informative. So this fraction of this portion of the script essentially determines the fraction of single positive cells that are, the fraction of cells in PIMO positive versus PIMO negative areas that each marker is single positive for. And again, we ultimately plot this as a bar plot. So if we take a look at figures and we sort it by date created, maybe let's view them as details. If we look at the very first figure that was generated, percent positive scores in PIMO positive areas, we can see exactly that. So this tells us in blue is the percentage of cells in the PIMO negative area that are positive for each of these markers. In orange, the percentage of cells in the PIMO positive areas that are positive for each of these 18 markers. And the number on the top corresponds to the fold enrichment. Essentially, what is the ratio of percent positive scoring in the positive to negative areas. Higher values effectively indicate an overexpression of the marker in positive areas, whereas lower values essentially indicate a underexpression in negative values. Or in other words, what's upregulated under hypoxia and what's downregulated under normoxia. Or that basically says the same thing. So now that we have this percent single positive chart being generated, why don't we, which is actually being generated here, we can also take a look at Violin plots. Now, Violin plots simply compare distributions. They don't depend on any threshold set by a pathologist or operator of this platform. So, for example, we can create a script that essentially iterates over each marker of interest and computes the Violin plots for all the cells in the negative versus positive areas. So if we take a look at one of the examples, such as alpha SMA, we can, again, see the distribution. Not only can we see the distribution, but we can perform statistical analyses comparing whether or not there's a difference in means across either one of these groups through essentially a standard t-test. Now, whether or not something is statistically significant or insignificant, that's still up for debate. I mean, most commonly people use a p-value of 0.05, but again, that's just an arbitrary cutoff. Different disciplines such as single-cell RNA sequencing will have much more stringent p-values, such as p-values in the order of 10 to the power of -100 or something extremely small. So as a result, we can get p-values of 10 to the power of -1, 10 to the power of -5, 8, 264. Or in some cases where the difference is so large, it is effectively a p-value that infinitesimally approaches 0. But again, this might be indicative that a conventional t-test might not be a suitable method for essentially determining differences between tens, if not hundreds of thousands of cells. So the great news about the script is that statistical analyses can basically be hot swapped, in and out, depending on whatever test is deemed appropriate by, say, a statistician. And of course, we can stitch all of these different Violin plots together into one nice plot. So, something like this would make an excellent figure for essentially determining whether or not there is a difference in distribution of marker intensity across multiple regions that have been identified throughout multiple IMC images. Because at this point, we're not really separating our experimental from control group yet. We're pooling all negative areas from both the experimental and the control groups, and all positive areas from all of the experimental and control groups together. Next up, we have select double positive combinations. So rarely could a cell ever be identified by a single marker. I mean, maybe an activated CD4 cell might be defined by being CD4 positive and, well, my memory is eluding me. I don't remember what the other marker was, but you might need a cell to be positive for more than one markers if you wanted to call it, say, a distinct cell type. Now, in conventional clustering techniques for cell classification, such as phenograph, you don't really have to worry about this. Instead, you look at heat maps of each cluster and assign a cell type according to this. The challenges relating to using clustering to identify cell types is that you kind of don't really have a way for a pathologist to basically make an informed classification for these cell types. And you also encounter issues if you have clusters that represent more than one cells, or you might have multiple clusters representing one specific cell type. So having the ability to define a cell explicitly as being positive for one or more markers gives this kind of tight grip on essentially controlling what is being classified as a cell that you are trying to measure. And so in this case, we can essentially identify cells that are double positive. In other words, CD68 and ICAM1 double positive in PIMO positive versus negative areas. And then, of course, we plot it over here. Now, pair plots are something that I've never really found a good use for, but it essentially is just a set of scatter plots comparing every possible marker combination to one another. And you can get some pretty hefty looking pair plots. You know, typically if you're working with a 4-channel fluorescent microscopy image, 4x4 grid is not that much, but when you're working with 18x18, you're getting somewhere close to 384 possible combinations. So if we take a look over here, the left axis is every single marker, single positive marker classification, as is the y-axis. And this is simply just another way of a high throughput method of marker-to-marker correlation evaluation. Points in blue are cells that came from a PIMO negative area. Points in red are cells that came from a PIMO positive area. And we can compute the Spearman's correlation coefficient only of cells in the PIMO negative area shown in blue, or only of cells present in the PIMO positive area shown in red or in orange, or independent of any region within the image, simply pooling all cells together and computing what their correlations are. We can do, again, the same Spearman correlation coefficient shown in black. So again, this is a great way, at least for studying the effects of tumor oxygenation on marker-marker or protein-protein associations. This will effectively tell us whether or not markers are upregulated together or downregulated together in response to variable oxygenation. And the diagonal of the plot simply compares distributions of individual markers, essentially the Violin plots that were previously shown. Now, pair plots, as you can tell, are fairly large, and you're going to be struggling to really find a way to integrate this into any kind of publication, let alone a presentation, without the ability to really kind of pan around and look for each and every marker combination that you're trying to find. Instead, we can generate heat maps that are simply colored by the Spearman correlation coefficient. So here we have a heat map comparing all markers correlated with one another, regardless if they fall in the PIMO positive or negative area. This one over here shows cells that only fall in a PIMO negative area, and this one over here shows cells that only fall in the PIMO positive area. And if we sort it by its correlation in PIMO, or with our hypoxia marker, referred to as PIMO, we can essentially identify what markers are upregulated or downregulated in response to this marker of oxygenation. So that essentially covers what the bulk of this code really does. It does a lot of Spearman correlation calculations, writings, little asterisks to signify statistical significance, plotting pair plots, saving pair plots, and of course we have the raw data. If you'd want to take a look at the p-values of each of these measurements, for example. So that essentially covers what can be done in terms of at least a first pass of analysis. Next up, we might want to perform some dimensionality reduction, not necessarily for clustering as we would with, say, Phenograph or things like Cytometry, but simply just to get an understanding of where do these data points lie on a reduced dimensionality space, to essentially see whether or not there are phenotypic similarities between individual data points across multiple groupings of samples. So, just as before, we load in our libraries, we read the data, specify, if we want to rename any columns, denote our measurements of interest, and we start setting up a UMAP. Of course, the first part of this UMAP is to, actually before that, I believe we want to remove any rows containing not a number. So Better safe than sorry, we have a little portion of the script that removes any values that would give us an error if trying to compute the UMAP function. Once we've computed the UMAP, we can essentially begin plotting the data with the X and Y axis corresponding to the UMAP dimensions. Something I also was looking into was seeing if we could improve UMAP reduction, or rather the clustering, not so much clustering, but I guess, yeah, reduction. If we started including other cell measurements, applying some kind of clustering such as phenograph clustering, as well as adding an additional dimension, maybe a third dimension towards visualizing reduced dimensionality data sets. So one example of a plot we can get is a UMAP colored by markers. Here's our UMAP, or let's start off with this one. We've colored our data points after UMAP dimensionality reduction based on whether or not a cell came from a patient that belonged to the experimental or control group. So now we're actually using some of the supplementary patient metadata that we've inserted when we were initially running the add metadata script. So we can also color the data points based on the region of interest that they came from, the specific image. So this can be used also for quality control. If we see one image that is drastically distinct from all others, we can always go back into the Keypad project, take a little closer look at it, see if the staining is appropriate, there isn't any non-specific binding, stuff like that. And of course, we can also color the data points as whether or not they came from a region belonging to a PIMO-positive or PIMO-negative area. So now when we go back towards looking at the UMAP colored by marker intensity distributions, we can see how are these regions, or rather UMAP areas, for lack of a better term, being colored by. So there's an alpha SMA positive area, there's a GFAP positive area, CD31, nestin, done for all of these different markers. Now, after we've computed the reduced dimensionality space, there's nothing to really stop us from performing clustering. The only reason I haven't done clustering yet is because given that we have this sort of semi-manual method of cell type assignment, there isn't really any new information that's gained from clustering unless we want to do a sort of unsupervised method of comparing differences across our various samples. So, it's kind of up to you, the user, if you want to actually implement this. And if you do, by all means, let me know. I'm more than happy to basically try this out on some of my own data sets. But going forward, specifically for IMC data, I was planning on using the phenograph clustering method, as that's widely considered a standard. However, for modalities such as codecs, there might be a slightly different standard. On a side note, another kind of great example of using cell clustering for cell type assignment would be Mike Nelson's Cytomap and Beck, I believe it's called. I might post a link to that in the video, but this is yet another method for going about assigning cell type classifications using dimensionality reduction and clustering approaches. Yeah, so on that note, I believe that wraps up at least the first pass of data visualization using Python. Now, I'll leave the remainder of this workshop open for discussions. I'm sure everyone here, or most people here, have some interest in analyzing IMC data towards Keypad. If you guys have any question with regards to maybe setting thresholds, classifying cells, choice of image segmentations, algorithms, I'm really open to discussion. And if you have any unique methods that you've also developed for analyzing high dimensionality data sets, I'm more than open to hearing about them. But on that note, I thank you for your time. I really appreciate all of you taking the time out of your busy schedule to take a gander at this workshop. I hope this was in some way, shape, or form informative to you. And feel free to reach out to me on the forums as well if you have any questions, or if you have trouble after this presentation when you actually try to run this workflow on your data set. Feel free to just tag me in any post. I'll try my best to respond. And yeah. And one last thing before I open everything up for discussion is the acknowledgements. So, I'd love to thank everyone who was involved in this project in some way, shape, or form. That includes my supervisor, Dr. Bradley Waters, as well as my unofficial supervisor, Dr. McKee, collaborators in the glioblastoma project from the Zadeh lab, Dr. Sula Mansouri and Dr. Gilroy Zadeh, my student, Dr. Miss Phoebe Lombard, who helped develop and write some of the Python scripts used here, as well as some of the unsung heroes who actually helped me tremendously with building some of the workflows and scripts, albeit in an indirect manner, I guess you could say. So Dr. Bankhead, amazing developer of QPath, extremely active on the QPath forums. The very first post I ever made on QPath was responded in less than 2 hours by Pete. Which immediately sold me on this platform. Mr. Mike Nelson, who's also doing some very exciting multiplex analyses and is a developer of the Cytomap and back for QPath. Research associate, the anonymous legend of the ImageSC forums, who's tirelessly provided, thousands of custom-made Groovy scripts in the blink of an eye. Dr. Sarah McArdle, who's also helped tremendously for developing different methods for segmentation, classification, and is a huge fan of the SLIC superpixel segmentation, which is also another thing that would be greatly useful in some of the multiplex analysis forums. And finally, everyone else who's contributed in some way, shape, or form through the ImageSC forums, who've been really instrumental in helping me throughout development of this pipeline, which I've ultimately used for this glioblastoma dataset, but dozens of other multiplexed IHC datasets from both IMC as well as Codex. So finally, on that note, thank you, and I'll open up the floor for discussion.